All right, hi everybody. We're starting chapter 7. We're going to take a look at the cell membrane structure and function. So first things first, we like to call a cell membrane lots of different things. So you'll hear us talk about the cell membrane, the phospholipid bilayer, or the plasma membrane. All of those mean the same thing. We're always talking about that boundary on the outside of the cell. And this boundary is really important. Not only does it protect the cell, but it serves as a selective barrier, so it controls what things can actually move in and out of cells. So you already heard me refer to it as a phospholipid bilayer. So it is made of phospholipids. So you have a phosphate group bonded onto some lipids, and then you've got proteins that run throughout the membrane. And so now we want to take a look at how are these materials arranged. So the phospholipid bilayer part is incredibly important. It's got a hydrophobic section in the middle. And you can see it's bilayer, meaning two layers. So you've got a top and a bottom layer here. This is the inside of the cell. This is the outside. This section in here is hydrophobic, which, like I said, is really important so that we don't melt in water. And then you've got the hydrophilic heads on either side that will interact with the cytoplasm and the extracellular matrix. These are two different models of membrane structure. We're going to take a look at this first one, the Davison-Danieli model first, and then we will take a look at B, which is the fluid mosaic model. So as you can see, just by looking at the pictures, they are pretty different. So both models have the phospholipid bilayer with the hydrophilic heads, set to the top and the bottom, and then the hydrophobic zone in the middle. What's different is where the proteins are located in these two models. So the Davison-Danieli model is from 1935. Again, phospholipid bilayer is there. However, this model had the proteins essentially coating the surfaces of the cell. And so this was called the sandwich model, with that phospholipid bilayer sandwiched in between the proteins. So evidence from this model came from some biochemical work, but you also had some transmission electron microscope pictures that showed the membrane as a double line, and so it was thought that that double line was from the protein coat sitting on top of and below that phospholipid bilayer. Problems with this model, though, were that not all cell membranes were the same, and there was just this idea of how were the proteins staying in place if they were just laying on top of and right below that phospholipid bilayer. So essentially this model was questioned and we had some more scientific evidence. In 1972, this is when we see the fluid mosaic model, and this is a new model that fits evidence that we have of what membranes look like, and this is a really good example of science as a process and how we question things that we think we know, come up with new ways of testing it, and gain more information. So the fluid mosaic model refers to the way that the phospholipids and proteins actually behave in the membrane. So the fluid part comes from that phospholipid bilayer. The molecules in that layer are actually not bonded together, so they're free to shift around, and actually they need to shift around. They need to stay fluid for the membranes to function properly. And so depending on what kind of cell you're looking at, there's actually different organisms and different types of cells have different ways of maintaining their fluidity. So plants will shift to having more unsaturated fatty acids. Like we talked about in class, the chains of those unsaturated fatty acids tend to have more kinks in them, and so that helps the cell membrane stay more fluid. In hibernating animals, you actually see an increase in the amount of cholesterol. So here we have a very rare example of how cells can maintain fluidity, and this is showing that you're having a flip-flop of the top and bottom phospholipids. This is just showing lateral movement, so that happens quite often. The flip-flop, though, is pretty rare. Down here, this is showing the unsaturated hydrocarbon chains with you can see they're all bent and kind of have those crazy shapes to them. So that's going to help this stay more fluid, whereas if it was all saturated, so again, like we talked about in class, that saturated fatty acids tend to have a much straighter shape to them. They're gonna, kind of solidify a little bit more than if you had unsaturated hydrocarbon chains. And then in C, you can just see that we've got some cholesterol molecules thrown into that membrane there. And so again, that's going to keep it more fluid. It sort of does the same job as these unsaturated hydrocarbon chains, just because you're not going to get these chains kind of laying really straight right next to each other because those cholesterol molecules are grouped in. Now the mosaic part of fluid mosaic model comes from the proteins. So these proteins actually float in a sea of phospholipids. When we say it's mosaic, like when you talk about art as a collage or a mosaic, that those proteins are they're gonna shift around and they're gonna change over time. So again, we have evidence for this. It comes from the transmission electron microscope pictures of fractured cell membranes. We'll take a look at that in a second. And we've also going to look at a cell fusion study. So what we have here is a cell frozen, and so you can see the plasma membrane. If you take a knife and very, very carefully chip away at it, you can actually chip the plasma membrane in half, and you can look at the two different sides. And when you do that, what you see is that you've got two different sides. So you've got the P face, which is the protoplasmic side, that's the cell side, and then the E face, which is the exterior face. And when you look at this, you can see that you've got proteins sort of stuck on either side of that bilayer there. The other example was a cell fusion study. So here the just very basic example is a mouse cell and a human cell with different colors to represent the different types of proteins within the membranes. If you look at this under a microscope and you study it, what you notice is that if you fuse these two cells, what you would expect if the membrane proteins didn't move was that, this half of the cell would be purple, this half of the cell would stay pink. But actually what happens is the proteins mix, and so this is what you would actually see after about an hour. So why do we have these proteins in cell membranes? They serve lots of jobs. They aid in transport. They help with enzymatic activity. Some stuff that we're going to talk about within this unit especially is that they're receptor sites for cell signaling. They help cells adhere to each other. They're used for cell-to-cell recognition and also for attachment to the cytoskeleton. So this is a picture you guys are familiar with. Again, we've got the phospholipid bilayer. Here you can see there's some cholesterol molecules throughout. This is the inside of the cell, so you've got the filaments of the cytoskeleton attached to some of these proteins within the cell membrane. And then on the outside, you have your extracellular fluid and your extracellular matrix. And so again, we've got some attachment to the cell. And now what I want you guys to look at is a couple different types of proteins. You can see that we've got these proteins that are sort of embedded within the cell membrane, and so we call those integral proteins. And then you also have some that stay on the within the cell. They don't actually go all the way through the cell membrane, and they're only located on the inside of that cell membrane. And those are your peripheral proteins. So this is just kind of giving you a breakdown. Again, transport, something we'll talk about very soon. Molecules can actually travel through the membrane proteins when they can't get through the phospholipid bilayer. So we've got some enzyme activity here. So these membrane proteins are an attachment site for these molecules, and they aid in a chemical reaction. Signal transduction we'll get to later on in this unit. Up here we have intercellular joining, so these proteins are actually helping two cells join together. Cell-to-cell recognition, so another sort of way that cells can talk to each other. And then attachment to the cytoskeleton and extracellular matrix, which down here you saw in the previous slide. And I already talked about this. We do have different types of membrane proteins. So integral proteins are actually inserted all the way in the phospholipid bilayer. They run throughout that cell membrane. And then there's the peripheral proteins, and these are not embedded in the phospholipid bilayer. They are instead attached to that membrane surface, but they are found only inside the cell. Okay, so a quick question. How is it that those integral proteins stick to the membrane or stay within the membrane? And it actually has to do with the solubility of their amino acids. So we talked about this way in the beginning of the year. when we talked about how amino acids have the different R groups and those R groups have certain properties, that is actually really important for these membrane proteins. So because the phospholipid bilayer has the two different sections, there's the hydrophilic section at the top and the very bottom, and then that whole middle area is hydrophobic. The proteins have to have both hydrophilic and hydrophobic areas as well. And so the hydrophilic amino acids are going to tend to be towards the outside of that cell membrane, and then again towards the inside of the cell, so they can interact with the hydrophilic part of the phospholipid bilayer. And then they're going to have hydrophobic R groups that sort of run within the phospholipid bilayer, very close to where those hydrophobic tails are. All right, something else that we want to talk about really quickly is that membranes are bifacial. So what that means is that they actually have an inside and an outside. So the phospholipid composition of the two layers is different. And proteins will have a specific orientation within the membrane. So they actually have a side that's supposed to face the outside of the cell towards the extracellular matrix, and a side that's supposed to face inside towards the cytoplasm. And we are actually going to look at a way to tell which way is which, and that is by looking at where the carbohydrates are. So the carbohydrates are going to be found only on the outer surface of the cell. And what you're looking at here is parts of the endomembrane system, and so this would be your rough ER, and then this would be the Golgi apparatus. And so you can see that the vesicles are forming, and actually even within the vesicles you have the carbohydrates on the inside. And they're on the inside here because when they fuse with the cell membrane and become part of that cell membrane, they are then going to be on the outside of the cell. So they're going to stay away from the cytoplasm. So the carbohydrates, when it's inside a vesicle or part of a vesicle, it's going to be towards the inside. And that is really helpful because then when that vesicle meets up with the cell membrane and opens up to release things to the outside of the cell, then the carbohydrates are on the right or the correct side of the cell membrane. So these membrane carbohydrates are branched oligosaccharides, and so again that's going to be, we had the monosaccharide, which was 1 sugar, disaccharide 2 sugars, then the polysaccharides where you're looking at lots and lots, oligosaccharides where you're going to have a few of those carbohydrates or a few of those sugars bonded together to form a kind of smaller polymer, but they are going to be the carbohydrates that you see on the cell membrane, and they're going to form, I know this looks like a lot, but it's glycophospholipid. So again, glyco, so that's going to tell you glucose, it's a carbohydrate. Then you've got the phospholipid, so you're looking at a lipid that has a phospho group bound to it. So this is going to be when you have a carbohydrate attached to one of the phospholipids from the membrane, or a glycoprotein is when you have the carbohydrate bound to the membrane proteins. And again, always going to be on the external surface of the cell. Here's your lecture question for tonight. So given the structure of the glycophospholipids and the glycoproteins and their location on the outside of the cell membrane, what do you think their function could be? All right, thanks guys.